So this is a presentation I have given for, or a modification of a presentation I have given to a variety of groups when we do manuscript and grant writing workshops and so some of you even on this call may have seen a version of this before. It's designed to be a mixture of best practices gleaned from other readings in the literature and other presentations I have seen, and then also just my own experience and my own kind of formula for what works for me and how I approach writing these sections. So I will generally try to call out areas where it's me being dogmatic or just presenting my own personal approach to things, and distinguish those from topics or things that I'm saying that I really think are best practices that should be important for the scientific integrity of a manuscript. This is geared towards manuscript writing more so than the grant writing. I think that we will probably have another didactic session on the method sections for grants later on in the program. There are only 16 slides in this presentation, and I think we have an hour or 45 minutes, so feel free to stop me as we go along. If you would like to ask a question, you can also ask questions at the end. All right, so do you see an advice? If anybody does not see the advice slide, will you let me know? Hearing none, I assume that the slides are advancing on your screen as well as on mine. So this is just advice that I have that many people do this already, but sometimes I come across people who don't do this. Just pick your target journal early in the process of writing your manuscript. Don't wait till the manuscript's done to figure out where you want to send it. This may change, your target journal may change as you write the manuscript, but it's good to start with a target journal in mind. I usually try to aim high for my first one, and then I have a second journal in mind if the first one, it gets rejected, which typically happens. So find similar articles in the target journal. Make sure that this journal publishes the types of article you're writing. And then you can also use those articles as a template for how to structure your manuscript. You can also find high profile articles in your field. This is twofold. There's twofold reason for doing this. One is it can also give you a template and examples for best practices for writing this type of article for your particular audience. And also you should probably be citing these articles in the manuscript that you submit. And so then follow their examples. So the analysis section, and this is geared more towards quantitative analyses, but I think qualitative research is also a highly structured methodology. And I think with some adjustments, this approach applies also to qualitative methods. I just don't work with those as much directly, so I don't talk about them. There should be a logical sequence to your methodology. The order you want to write about your analyses in the order in which they're introduced in your aims. In a manuscript, your aims is really more your introduction section. They're kind of analogous to the aims page from a grant. And in fact, I typically take my aims page and that becomes the first draft of my introduction when I'm writing a manuscript. So the analyses should be addressing the scientific questions that you raise in the introduction. They should proceed from simple to complex, and they should be presented in the order in which they were conducted. So you describe the analyses in the order that you would conduct them. You want to also distinguish a priori analyses from post hoc. This seems very obvious, but it's there's a lot of articles that get published even in good journals where it's not always clear what analyses were planned before they saw the data and what analyses were done after they saw the data. And those are two very different scientific products. And so it's important to make sure that you are clarifying that and being transparent about what you plan to do before you knew your outcomes and what you did after. So do the analyses you originally planned, even if the results aren't what you hope to see, or if you had problems with the data collection, I would still do those analyses, present your results, and discuss the limitations. And then if for whatever reason you decided to add other analyses on after the fact, that's not bad. You just need to be very transparent about it. So for the analysis methods, I know some people on this cohort of RCMAR scientists are methodologists themselves. And so there's two kinds of first authors, those who are the methodologists and those who are not. Even if you're not doing your statistical analysis, if you're an epidemiologist or a statistician or a quantitative data scientist of some type, then your role as first author will be a little bit different in how you interact with your data analysis and method support if you're not doing it yourself. But really, even if you're a first author who's not a methodologist, somebody who doesn't have more than maybe rudimentary statistical training and skills, you still should be intimately involved in the writing of your analysis section for your paper. If you're the first author, you want to make sure that this analysis section reflects what you wanted to do and that it's done correctly. So first of all, you can keep it simple. Analysis methods can be complex, but the writing about them doesn't need to be confusing. So, and then also just the analyses themselves, start simple. Complex methods increase the risk of errors. The farther you get from your data, the more parameters, the more assumptions you have to layer on to your analyses, the more kind of wiggle room there is in interpretation. The looser your interpretation often has to be because there's more caveats, more conditions on your interpretations. If you're doing a large randomized controlled trial and you can do a t-test, we always say, of course, that that's the ideal situation, if that's all you need. Rarely is that all that we need because our data aren't perfect large randomized controlled trials generally. So you use more complicated methods to address what might be considered as limitations in the data set. And that's a simplistic way of putting it. But if you're talking with a statistician and it feels like your analysis section is getting complicated and confusing, at least back it up to simple methods. Make sure you understand why those simple methods aren't going to be enough before you move on to the complex approach. I've reviewed manuscripts for journals where they launched straight into a complex analysis without doing the simple analyses first. So I had no context for interpreting those complex methods, and that can be a problem too. This middle point is something that I am a real stickler for. I think it's very, very important. Your scientific question should drive the analysis plan, and the analysis plan should drive the results. It should not be the other way around. This seems really obvious, but it can be surprisingly difficult to do. And partly that's because of the pressure to publish around so-called significant findings. But going the other direction, which is what I often call having the tail wag the scientific dog, is a real problem in terms of replication and publication bias in our public health fields. So your scientific question guides the analysis plan, and that guides the presentation of results. This actually means that you can write everything up to the analysis and including the analysis section before you've done any data analysis. That's what I always recommend. Not that any of us want to compromise the directionality of our science, but we're humans and we're subject to these pressures. And we get excited about things we find that maybe weren't exactly what we were looking for. And it can be very hard. It can become quite a gray area if you're writing the introduction and the methods after you know the results of your analysis. And then adjust for confounders thoughtfully. That's another common pitfall I see in statistical analyses, both primary data collection and secondary data analyses, where people kind of throw a kitchen sink of confounders and do a big adjustment without really thoughtfully thinking about the role of each variable in relation to the exposure or the independent variable of interest and the outcome. A big problem there is when you're adjusting, if you do a multivariate adjusted model and you say every association here is adjusted for the presence of all the others in the model, that's a problem because when you adjust for downstream effects, so you have some exposure which causes something which then causes the outcome, so kind of a mediation approach. Adjusting for that as a confounder is not appropriate in that particular exposure outcome relationship. So if something is a confounder, so it just gets very muddy. So if you have a big multivariate adjusted model, I usually avoid doing that for your primary analysis because it gets very hard to interpret many different associations in those models. Okay, the analysis plan should not actually have results in it. The exception is that sometimes we sort of foreground results if they influence the analysis. For instance, if there was some unexpected problem with the data or something that happened that triggered the need for another type of analytic approach, sometimes we might foreground that in the analysis. Write for your audience. And this is another area where I feel this is kind of my dogma, my best practices. But who's your audience? Are they statisticians? If you're writing for statistics in medicine or biometrica, then you can get into the nitty on parameters and statistics and jargon. But most of us are writing for a public health audience that are not necessarily theoretical statisticians. And so often you can keep it simple when you're performing, when you're describing the analyses and the results. We're often looking for group differences. You know, we want confidence intervals. The statistics themselves, those actual numbers, they can be used to help contextualize these differences and these confidence intervals, but they're not necessarily the goal for the reporting, depending again on who your audience is. What do they care about? When you're writing an analysis plan or reading an analysis plan that your statistician has written, you want to make sure it links back to the a priori scientific question. When I'm writing an analysis plan, I often will include in the early drafts an explicit statement of what scientific question each part of the analysis is addressing. And maybe I take that out before I publish the manuscript, but it helps me make sure that I'm linking back to my a priori scientific questions, and I'm keeping that parallel construction of presenting everything in the order in which it was originally described. If you have a priori decision thresholds, make sure you use those. And then you want to describe power in the analysis plan only if it's applicable. So power isn't always applicable when presenting results of an analysis. For randomized controlled trials, it is applicable and it's required as part of a consort, the consort process. But for secondary data analysis, power, once the data have been collected, power really, for the actual manuscript itself, becomes pretty irrelevant. So although in some situations you might allude to power, or there can be exceptions about when you should be talking about power, it's rarely meaningful to include a power calculation for a secondary data analysis. And you should always say what statistical software package you used. Sometimes different packages do things slightly differently. They make different assumptions. They have different defaults. So it can be useful to some of your readers to know what the software was that you used. All right, so this slide talks about collaborating with your statistician. And again, this is really for people who are not themselves the statisticians on your first authored papers. You want to involve your statistician early. Everybody on this session who has performed a support role as a statistician has had the experience of somebody coming to them very late in the process with all the data already collected, and they just want you to say whether something was significant, and so that they can publish the paper and move on. And that's not an effective way to work with your statistician, and it's not a very good way to make meaningful contributions to the scientific literature. So involve the statistician early. Make sure they understand the context of the research, make sure they understand the question that you're asking, and they understand how the data will answer that question. And that again seems really obvious, but often you're going to be working with a statistician who's not an expert in your field. And so they're not going to be overly familiar with the methods or the variables in your data. So make sure that that's very clear to them. If there are scoring packages or kind of ways of summarizing certain instruments, make sure that they know that. And that's for, I think sometimes people want the statistician to go out and find that information, and they certainly can, depending on, the arrangement you have in your workplace. But the problem is that they're not experts, and they are often not going to do it right. So we can go out and look at measures or look at ways of scoring things, but we can't guarantee that we're using the best one or the most recent or the one that your reviewers who are experts in the field will expect. So it's good to at least be involved with that process. Your statistician should write the analysis plan before conducting the analysis. I never do the analysis and send it off and then after the fact write up the analysis plan. I write up the plan first. I make sure it's very clear what questions I'm answering, and I send it to the people I'm working with, and I say, these are the questions we're answering, confirm that these are the ones that you wanted me to do for you. make sure that you understand the analysis plan. This is really important, especially if you don't consider yourself a methodologist. As long as you're an educated public health researcher, you should be able to understand your analytic plan that your statistician produces for you. It doesn't mean that you have to understand the nuances of every method that they use. There may be words or terms in there that you're not fully familiar with, but you should at least understand the point of having them in the paper. So for instance, if you're working with someone who's doing a Monte Carlo method for an analysis that you're working on, you don't necessarily have to know how that method is implemented, but you need to know why they're using it. Why was it appropriate for this particular project? I think, and that's especially important if you're the first author, just assume that your audience is going to be people like you. And so for some examples that I've come up upon are just why are they doing things to the data? So often I read papers where they log transformed non-normal data. Well, that's actually only necessary in very specific situations. It's not a default approach you have to do anytime you have non-normal continuous data. And every time you log transform, you're moving away from the crude data. You're making it harder to interpret these differences or these findings. And so if you see, for instance, a log transformation, make sure it's very clear why that was done. Again, simply having non-normal data all by itself is not enough of a reason to log transform. You need to be violating some assumption of a model or something like that that requires log transformation to get closer to those assumptions. And then also things like statistics. What does an F test really mean? Every time I read about ANOVA results and they give me a bunch of F tests, but they don't talk to me about group differences between the factors that they're studying, it's always really hard to interpret what does that really mean for public health or for clinical research. So make sure that you understand it and you understand what the results mean in terms of the questions you're trying to answer. I usually plan tables and figures before doing the analysis. I find that especially when I'm not doing the analysis myself, even though it helps me communicate with my statistician. I say, these are what I'm thinking of, or I ask them to send me blank tables to show me how they're thinking to present the results. Now that often changes as you go through the paper. You realize there's maybe better ways to present your findings, or your reviewers request different ways to present your findings, and that's fine. But I usually start off. So I do a whole lot before I ever really touch the data themselves. The results section, the structure, again, this is going to be very straightforward for many people, but I find it useful to at least review it because sometimes we have people who primarily publish in, say, sociology or other fields where it's a little bit different than in most of the biomedical literature. So the descriptive table should be table 1. The purpose of table 1 is just to show your readers the data you have. It's not to make any inference. It's not to do any testing. It's just to say, here's the data we worked with. So there shouldn't be any p-values in your table. P-values imply null hypothesis significance testing, and that's not what table 1 is for. It's just descriptive. You can still talk about patterns you see in the table, but you're just describing them qualitatively. You're describing your impression, what you think is important in that table. You're not doing official tests. If you have a consort diagram, if you're doing a randomized controlled trial, that's also a descriptive part of the paper. So that would be early in your results section. Then usually you would be doing some kind of crude analyses before all the fancy adjustments. If you have a really simple big randomized controlled trial, this might be all you have to do. You might just do your descriptive study, your descriptive table, and your simple analytic table, and that might be enough. And that's fine. That's great, actually. So table 2 would do that typically. The unadjusted results, maybe with some very simple stratification. I always give confidence intervals. I tend to not give p-values because I think people tend to focus on them more than is appropriate, but I always give confidence intervals. So that would be table 2. And then if you do more complex analyses, or you do sensitivity analyses or post hoc analyses that maybe weren't planned ahead of time, those would be tables 3 and beyond. Typically, this is just a very simple structure. It can be very different when you actually get into writing your paper. Things change depending on the context. But here's where I would do maybe anything that adjusted for missing data, or things that adjusted for interactions or confounding or things like that. I would put those in tables 3 and beyond. Sensitivity analyses, sometimes you present those in tables, sometimes you just say in the text that you did them and they didn't really change the results, so you don't have to report those. And then figures as useful. Sometimes all you need is a table, sometimes a figure is more effective than a table for presenting your results. So here's the really important stuff. These are the pitfalls that I see surprisingly often in manuscripts that I review. You don't want any new analyses suddenly jumping out at your readers. Every analysis that you present in results should have been discussed in the analysis plan. You don't want any new variables. That all the variables you use in your analysis and presenting your results should have been described in the measure section before you get to this section, to this part. You want all your pre-planned analyses, all your primary analyses to be also presented in your tables and figures, because lots of readers aren't going to look at the text of your paper. They're going to jump straight to the visuals. So anything that was pre-planned should be in the tables and figures. And then the other thing that I think there's I think quite a bit of variation in how people approach the text for the results section. Some people restate every number that's in the table or figures in the text. I don't recommend that you do this. Biomedical journals like to keep things short, and people can look at the tables and see the actual numbers for all these different, all the different descriptive cells in table 1, for instance. You don't have to go through and reiterate what everything is. If there's something that you think is very interesting, especially something that you're going to refer back to when you're in the discussion section, call that out, or you call out the things that are pertinent to your main scientific questions. You don't need to layer on interpretation, but you can just call out findings that you want to make sure your readers notice. That's what I put into the text. You definitely don't need to restate everything. Again, keep it simple. Just give the statistics that are useful. Most statistical software packages spit out a whole lot of stuff, and some statisticians like to present a lot of it for various reasons. Some think that you should present all of it for completeness. Some just aren't sure what to really present. But keep it simple. Only give the statistics that are useful. What are the statistics that you need to answer your questions, and understand what your findings mean in terms of your variables and your specific study and question that you're trying to answer. Again, make sure that they're presented in the same order in which they were described in the analysis, which is in the same order in which they were introduced as scientific questions. And don't put all your eggs in the p-value basket. P-values are one tool. They can be a useful tool, but they're not the tool for determining whether something is important or whether there is an association that we should pay attention to. So you can mention p-values, but just because you have a p-value above 0.05 doesn't mean that's a null study. You can have compelling findings that support an association that is meaningful from a clinical or public health perspective with a p-value over 0.05. So that's why I would say use confidence intervals and work with your statistician and think about what these results mean as you describe the findings for your study. So you can say what they mean, but again, you don't interpret the results. You don't put any really subjective interpretation onto the results. That happens in the discussion. Okay, table 1. This is going back to what I said earlier, kind of restating it, no p-values in table 1. If you're publishing a paper and your reviewers request p-values in table 1, then you have a decision to make. I usually try to push back against it, but there are times where I just give in and give them the p-values. It depends on the situation. Again, in tables 2 and 3, and when you're presenting the findings from your inferential analyses, just give the statistics that are useful for answering your question. I always harp on the F-test. I don't think anyone really knows what an F-test means. Rich knows, but I don't think anyone else does. And then make sure that you're presenting all the results that are needed to understand the main findings. This is why it's really important to present those simple analyses with the mostly just kind of the crude data in table 2, because it gives your readers the information they need to maybe question the findings in your more complicated analyses. So when you're looking at a paper and they jump straight from the descriptive table at baseline, say, to the complicated analysis findings, but they don't give you the basic data that you need to construct some of your own, say, 2x2 tables to look at how those outcomes seem to have maybe been different or not so different between groups of interest. That's a flaw in your paper when you don't present that information. So make sure that you give people everything they need to understand what you're presenting, and also to come up with their own conclusions about whether they agree with what you're presenting. Tables, the conventions for biomedical literature, they're simple, but I find that reviewers get persnickety sometimes when people don't dot the I's and cross the T's, so to speak. So I throw this in a table, the expository title, so table 1, and then the description of what the table is, that comes above the table, and it should stand alone from the text. You want to avoid abbreviations in your tables. If you need an abbreviation at the bottom in a footnote, you need to define it, even if it's been defined in the text. Again, many readers aren't actually reading your paper in terms of what the words that you wrote. They're looking at the tables and the conclusion and maybe the abstract. Another thing I feel pretty strongly about is using the fewest digits possible to tell the story. And what do I mean by that? I mean, you often don't need to use anything to the right of the decimal point to convey what you're trying to convey to your reader. When you do need to use significant digits, use as few as possible, because every significant digit you add in there is a cognitive burden to your readers. Too many significant digits and too many statistics makes my eyes glaze over, and I start scanning, and I don't really pay attention to what's being presented by the authors of this, work they've done. So my rule of thumb is you use one digit, you go one digit smaller than the magnitude of the differences you're working with. So what I mean by that is round to the ones digit if you're looking at differences of, say, 10 or more. So if you're looking at 72% versus 64%, that's all you need. You don't need 72.345 versus 64.8910 or 890. That doesn't matter. Those don't give you any useful additional information. They clutter your paper and they make it harder for your reviewers to follow your work. Round to the 10th if you're looking at differences of one digit kind of or more in the ones column. So 1.2 versus 3.6, 1.23 versus 3.64 probably isn't going to add anything to your readers, for your readers. Okay, so just keep it simple, keep it concise, and keeping it visually tidy, I think really does make a difference when you're presenting your findings. Figures, for figures, the expository table title goes below the figure. I said table there, but it should be figure. So I don't know exactly why this is the convention, but for tables, it goes on top. For figures, it goes on below. Some reviewers get cranky about that. So I would just recommend doing it the way that they're going to expect it. Others won't care. I only use figures if they're useful for visually conveying something important. I like figures. I think often they're a lot nicer than tables, but they tend to take up more space to present, less information, which can be fine. And it can be really important for highlighting something that might otherwise get lost in a table, but just make sure that it's useful because they do take up a lot of space, and sometimes your articles will get a more welcome reception if they don't have a lot of figures, making them longer. When you do create tables, make sure that they can be fully understood when they're just printed out in black and white. Some journals have requirements around this, some don't, but it's fine to use color, I think, especially these days, since lots of us are looking at our papers. We look at manuscripts online on the computer, and so we see color. But yeah, you want to also make sure that if someone prints it out on a black and white copier, they can interpret that table. So that's part of a hard copy edit. I always print it out and make on black and white and make sure that it looks okay. So I am just very briefly going to give you the approach that I use to write the discussion, because it's pertinent to the construction of the analysis plan and the results, because the discussion is where you talk about all those things. So you've hopefully set yourself up for your readers to have the context that they need and the information they need to digest your discussion. So I always start by restating the original question and the key findings. Don't cherry pick significant results that were not the original primary question. You can talk about those surprising findings later on, but just make sure you restate the original question, restate the key findings, interpret them in comparison to other research, and then I discuss secondary findings. If we did post hoc analyses, if we did exploratory findings, if there were unexpected results that either contradict other studies or that we weren't really looking for but jumped out at us, you can mention those, but don't overinterpret them. Treat them as hypothesis generating, which is what they should be. If there's any special considerations, then I talk about that. And then in your limitations section, you talk about, you would definitely want to mention your limitations when you're working with your statistician on the analysis. That's part of understanding why they're doing certain things in the analysis. That should start to inform your limitations section. Respond to each limitation, but don't just list it. Don't just say, here's a limitation. Explain after that, give a sentence that says, why is this still worth publishing? Why is this still a useful contribution to the science? Every project has limitations, and the question is, does it still matter? And then in the last paragraph, again, summarize your key takeaways. Anything that you're summarizing here should have been content that you called out in the results section. Your key findings could have been should be things you called out in the results section. You didn't have to dwell on them in the results section, but I would always mention them so that when you come back to them in the discussion, your reviewers or your readers are more likely to have noticed them when they were reading the discussion if they read your text. And then you just end with your final takeaway. The last paragraph, for the people who just skim the abstract, look at the tables and figures and read the last paragraph, that's going to be the last thing they see. These are some resources that you can look at. These are some of the things I looked at when I was working on this presentation. There's lots of other guides out there for science writing and scientific writing. And that's it. I had to, yeah, I found this. I liked this table. I think it's useful. We all strive to convey complex subject matter with easy to understand writing, but often we also see simple subject matter combined with writing that's hard to understand. I had to do a little censoring to what they called it, but that would be not such a strong contribution to science. Anyway, are there any questions? Are there any thoughts? I hope that even for those of you who are experienced paper writers, that this gave you at least some ideas, or sometimes just ideas for what you don't want to do, because something that works for me does not work well for you, and that's fine too.
Hi, thank you, Clemma, for this amazing presentation. I wanted to ask you, is there any chance you could share the slides with us?
Oh, yes, I should have said, I think Kira usually sends these out after, or else we can make them available on the website. But yeah, yes, absolutely.
I have a copy. I will be sending them out to everyone.
Yeah, you can have these.
Okay, great. And I wanted to ask you a question about something you said in the results section. And this is something that I personally struggle a little bit with, which is this idea that you need to say what the results mean, but you don't want to interpret them. Can you elaborate on that a little bit?
Yes, and I knew when I wrote that can be a gray area, because there is subjectivity in the results section. So I try not to say too much. I try not to differentiate too much between objective and subjective writing, because I believe that all of the science we do is subjective. How we ask questions and what questions we ask are subjective. So there is subjectivity in your results section in terms of what you call out. So if I'm looking at table 1, there may be a number of things that I could focus on for potential differences between some groups in my study, but there's a few that I think are the most important in terms of how we then interpret the inferential analyses later on. Those are the ones I'm going to call out, because I know my readers can look at the table, they can see all these differences for themselves. So I'm basically telling them, here's what I think was the most important takeaway from this table. So I'm putting my own subjective interpretation on it in that way. This is what I think you need to focus on when you look at this table. I'm not going into detail about the why, about the implications of that. about what that means for the field or the study, that I'll do in the discussion. Does that answer your question, Anna?
Yeah, absolutely. Thank you.
Okay.
Hi, Clemma. I found it interesting that you mentioned not to include p-values on table 1. And, because some journals, they have a very limited number of tables. And sometimes when you have like 2 or 3 groups, it seems just easier to include the between group differences with p-values. So I don't know, do you think there are exceptions about it, or?
I think if the purpose of the table is to describe the sample that you worked with, I really can't think of any time when I think p-values would be meaningful, because the purpose of that table is not to formally test for differences between groups. You know, that's a different kind of question. It's just to show your readers what the data were that you were working with. That's my own strong personal opinion, and I fully realize that many people feel just as strongly in the other direction. So I definitely acknowledge that. But you know, to me, it's a inferential, p-values are an inferential statistical tool. And so unless we're asking an inferential statistical question, I don't think p-values are appropriate. But as you said, some journals have very limited, maybe they have a very limited number of tables that they allow. And so you can kind of combine inferential and descriptive statistics. Maybe that makes sense for a project. So I'm not saying I would never do it. If I did it though, I would say why I did it. And I would still try to make sure that I was differentiating between the descriptive evaluation and the inferential evaluation in my interpretations.
Okay, it makes sense. Thank you.
I knew when I said that there would be people that went, what? So, because it's very also field specific. I know that there are fields where it's really kind of a big deal to not put the p-values in table 1.
So Clemma, thank you. This was very helpful in how to structure a paper. And yes, I definitely want a copy of the slides. And I was just wondering if you could comment on if you have a study with multiple measures, but there's only key ones that you're actually looking at your specific aims. I think, you know, keep it simple, only give statistics that are useful. But if you introduce these measures in the methods, do you still address them in the results, or do you see what I'm saying? If they're peripheral to your specific aims, but they're sort of part of the study. Do you see what I'm trying to say?
So you mean, for instance, maybe you have a single exposure of interest,
yeah,
but you're adjusting for variables, and
yes,
when you report that, those have parameters associated with them
yeah,
that people might
yes,
want to care about. So
yeah.
I address that in a couple of different ways. Usually what I try to do is, you know, only report statistics that are really relevant to the question I'm asking. So often if I'm doing a whole multivariate adjusted model, I don't report that whole table with all of those parameters and coefficients for every variable. I'll just show how those primary point estimates that I care about change. So I'll say, here's the point estimate and confidence intervals for the unadjusted model, you know, and here's the point estimate and confidence intervals for this exposure that I'm interested in after adjusting for these other variables. I don't necessarily give the point estimate and confidence interval for each of the additional variables, because those aren't the question, right? Yeah. That association is not the question I'm trying to answer. Yeah. Now that's more for a very focused inferential question. Sometimes our papers are intended to look at a bunch of different things, and that's a more hypothesis generating paper, right? That's not a narrowly focused paper, scientific question, and that's fine too. So then you might present a whole lot of different parameters with confidence intervals, coefficients and confidence intervals, and that's okay too. I would just make sure that you're interpreting it appropriately, right? So it's just to be, when you're presenting a whole lot of association estimates for lots of different potential exposures for an outcome, it's very easy to overinterpret significant findings, statistically significant findings, so to speak. So again, I think it's just really important to be careful to appropriately interpret those as more exploratory or hypothesis generating, as opposed to, oh, hey, look what we found, and then focus your whole, you know, don't focus your whole discussion on this one statistically significant finding, because there's a strong chance that it's really, you know, it may not hold up in future research. So sometimes though, if you are doing a focused analysis where you really have just one or two, say, exposures of interest, but you're adjusting for things, maybe it's a standard for this journal or your reviewers requested that you provide those coefficients. And so then again, you just have a decision to make, whether you want to push back against that or maybe offer to publish it as a supplemental table to try to minimize the risk that people are going to overinterpret these findings that weren't part of your a priori question, or you just present them. There may be interesting hypothesis generating, kind of ancillary findings that you should call out, and that's fine. Just again, treat them as such. Just say, hey, when we did this and we did this adjustment, we found this strange thing, and don't focus too much on it, but you can acknowledge it and say, it'd be really interesting if a future study, checked this out more carefully to see if this holds up in other data or other contexts. So I think it's really keeping your eye on the prize. What was the a priori question? What did you need to do to answer that? And then what else is there that you want to talk about? But just make sure that it's very clear, especially if there are things that came up more post hoc. Just make sure that it's clear that you're very transparent about what's what.
Clemma, if I could just chime in too, I think this is great, and I think it gives you a great framework, for doing your statistics. And Clemma mentioned this, if you are eventually going to publish in a biomedical journal versus a social science journal, everything that we say may seem diametrically opposed to what the journal's telling you they want, right? So the idea is this is how you do it correctly and do it complete. But when you're actually writing, when you're actually making the paper, certain journals will have very specific requirements of the way they want things, and other journals will have entirely different requirements. So you want to do your analyses and really kind of write out your tables and your paper. And then I guess what a lot of us do is look at what the results are and say, okay, this is a high profile result, I'm going to go for this journal. And then you read the instructions to the journal, and then you find out that, well, they're only allowing you 2 tables and 1 figure. Well, now I really have to decide what I'm going to show and what I'm not going to show. And that's where you get into the challenge, like what Clemma was saying, was normally you would have a table 1 that's purely descriptive, but now you can't really, because you're only allowed 2 tables and 3,500 words. So I have to try to get as much information into a table, which seems, again, almost a contradiction to the lesson you just had, right? But it's not. The idea is create the tables and understand what you need to do, and then try to format it for that journal. And sometimes I choose journals, or sometimes I don't choose a journal because there's just no way I can meet that format, right? I recently published a paper on mindfulness in the Journal of Alzheimer's Disease, which does not have a page limitation. But I originally wanted to put it in the Journal of Alzheimer's and Prevention, which allows 30 references, 2 tables, 1 figure, and 2,500 words. And there was just like no way I could write a paper on mindfulness and dementia in 2,500 words with 2 tables. So no matter what you want to do, you kind of have to go back and look and say, this is the data that I have, and this is what I'd love to present, and then I try to find a journal that helps me express that. So, sometimes there are competing interests, but you should always let the data drive you, as opposed to the other way around.
Yeah, thanks, Jim. And it's good to reiterate that this is a general approach to how I take to writing papers, and that I work with the people I mentor. I generally guide them towards this approach, but it's more of a template. And then you have to build the framework around it based on your field and the journals that you're trying to publish in. The things that I think are consistent across any field and any journal are just the kind of scientific order of operations, and just the importance of being transparent, letting your scientific question guide the analysis rather than the other way around, and the results rather than the other way around. And again, just being transparent about what did you intend to do, what did you do, and then how do you interpret those findings appropriately. And then the exact kind of way that presents is going to depend a lot on the journal that you're trying to work with.
Yeah, and the last thing I would just chime in is, if you've largely been a biomedical science paper, journal writer, and you try to write for a social science journal, it's quite a shock to sort of change, the way you present things. And if you've traditionally been a social science journal publisher, writer, and then you try to write for a biomedical journal, it seems, sometimes they seem, again, very, very different. So, you know, let the science drive you and then try to figure out what, you know, what's your target audience and the like. So I'm going off the analysis side, but part of the analysis at the end is that you'd want to be able to present this in a format that people are going to read. And so if, you know, if you're a sociologist, you know, you want to publish in journals that are high impact in your field, right? And so the structure of those articles are very, very different than if you publish in neurology, right? Which allows you a 500-word introduction, 5 tables, 50 references, 3,000 word limit, not including references, and tends to be really terse in its discussion. And you know, having published a lot of papers in neurology, no reviewer has ever asked me for confidence intervals. Okay. On the other hand, publishing in another journal, you know, that's what they want to see, because that gives you a much better sense of what the data really looks like. So again, as someone who's published a lot of papers, you know, do your analysis, look at everything in front of you, and then think about what's the best format to present your research. So, you know, the second part of this presentation is, now what do I do once I've done all this? But first you have to do it, and this is the way to do it right. Or this is a template for doing it right. Shouldn't tell you that's the way you have to do it, but it's a really good guideline for how to set things up and be successful.
Thanks, Jim. Are there any other questions?
I'll ask, I don't know if it's a question or really just, well, I guess it's a question and a comment. You covered quantitative way to present data. You know, a lot of folks in our programs tend to have qualitative data, which I wouldn't ask you how to present. But I think the challenging part is when you have qualitative and quantitative, and you don't want to just present them as 2 totally separate things. So I wonder, you know, you're very familiar with this since a lot of people do this. I'm wondering if you have any suggestions, because it's not a matter of how you present the data, it's really in the interpretation part of, you know, the paper. So that's one question or point. And the other point is, and this may come up during our review of the, you know, people's products, and this is really basic. And we have had 2-hour discussions about how do you actually set a table up? What goes across the top and what goes across, you know, the X and Y axes? Because how you set it up is critically important to both your, you know, your aims or the goals of the paper, and the information that you know, transmit. And I think a lot of times people just set them up sort of without thinking about that, what goes across the X and the Y axes. So just wondered about your comments on that.
Sure. So first of all, for the tables, yeah, I intentionally didn't give examples of that in this because I felt like you'd almost need a whole different presentation to discuss how you present tables. And because that's something that each scholar should be working on with either their statistics member or their primary mentors on your team, because it is also going to be largely dependent on your project and what you're doing in your analysis. But Deidre's very right that you need to be very thoughtful about how you construct your tables. And that's another reason why I like to create blank tables before I ever do an analysis, especially if I'm working with somebody else to implement the quantitative data analyses, so that we're thinking about that right off the bat. How are we going to be presenting this? You know, what's the information we need to see, and how should it be arranged? And also, again, that's another place where finding an example of a very similar manuscript in your target journal can be very useful, because you can look and see how they presented it. You know, were they looking at things by rows? Were they looking at them by columns? Did they, you know, split up the descriptive table based on the outcome or based on the initial exposure or a variable of interest? You know, so you can just look at how it's done by other people in that journal or that field. In terms of the qualitative and quantitative analyses, when they're presented together, I think that's really tricky, because for one thing, as Jim alluded to, often you have word limits, and it can get very difficult to present both qualitative findings and quantitative findings from a project in a single manuscript. I feel like there's a number of times where I've worked with people where we started trying to do that, and then we realized that we really had to split them out, simply because we couldn't do them justice in just one paper. But often when we're doing mixed methods research, where the qualitative and quantitative activities are really more intimately integrated, then you can get into this sort of paradoxical situation where it's hard to fit everything into the word limits, but it's also really hard to separate them out because they were inherently, mixed together. And so I think, you just have to try to work with your team and figure out what you're going to do in that situation. One option might be, as Jim mentioned, you look for a journal that doesn't have limits. I had a paper where we had, I think, 7 tables, and we really needed those 7 tables to describe our findings, to present our findings. And we went to the Journal of the American Heart Association, which doesn't have a table limit. And so that was where we published that paper. So, you can look for a journal that allows long papers. You can also, consider, can you break it up, first into maybe a qualitative paper or quantitative paper, and then pull from that. And so that the integrated piece happens in the second paper when you're, discussing the intervention you created based on the qualitative work, or, there's a lot of different ways you could approach it. I'm not a qualitative researcher, so I don't know as much about how to present qualitative findings. But I think the times when I think I have worked on papers that did that, we tended to have to kind of silo them, right? So we tended to sort of do all the methods and results for the qualitative piece first, say, and then all the methods and results for the quantitative piece, with discussion about how they were integrated. That's been the approach I think that we've used in the past, but really I can only think of a handful of papers where we really did present those results together. Usually I feel like, well, a lot of the work we do is we use focus groups or qualitative methods to, say, adapt an intervention, and then we test it. And often, those are 2 separate papers.
Okay. Spiro, hold on a second here. Spiro has a couple of comments too.
First off, I think it's important to recognize that there's typically an understanding in the general consciousness that qualitative and quantitative methods are independent and at best parallel and sometimes intersecting in their relative contributions to the nature of the question. They can differ in terms of their relative contribution to articulating a research question as well as answering a question. And I think one of the main false assumptions that people make is that qualitative research is not subject to the same level of rigor and specificity as quantitative research and the assumptions and methods that underpin it. And as somebody who focuses on both, but particularly trained with respect to qualitative investigative methods, I have the same expectations as somebody such as Clemma in regard to more quantitative approaches about the ability to specify sampling frame, the sampling strategy, the representation issues, et cetera, et cetera. I find one of the most common mistakes is that authors who depend upon qualitative investigative methods if they're not that experienced, they'll assume many things are self-evident or don't require the same level of specification. And that will lead one to be very seriously critiqued. So those are some of my initial thoughts. And, when people talk about qualitative investigative methods, Clemma may open by saying focus groups. Well, I got to tell you, focus groups are an original business industry marketing technique that found a place within the social and behavioral sciences. And while the same notions of rigor and systematic presentation and implementation apply to focus groups, If you're talking to truly qualitative investigators like anthropologists or some sociologists, we kind of smile to ourselves about the equation of focus groups and qualitative research. So when we talk about qualitative research, focus groups are a very small, minor subset of those investigative methods. We're thinking about participant observation, semi-structured key informant interviews. We're thinking about ethnographic work, etc. And there are volumes in anthropology and sociology about these particular approaches. And they also then lead directly to a variety of different kinds of analytic procedures that need to be undertaken, described, and reported in equally rigorous fashion. So I just want to make sure that we leave this discussion with an awareness that the same kinds of standards can and should be applied to qualitative investigative methods as their quantitative counterparts.
Absolutely. Absolutely. And I think, again, like I said in the beginning, the general approach of outline your questions, outline your methodology, write your plan, and then present the results, that applies to any type of research methodology, be it quantitative or qualitative. And absolutely, qualitative research should be approached as rigorously as quantitative research. And then I guess as Spiro was talking, and I was thinking about Deidre's question about how do you present studies that employ both. I guess I would say you start at the beginning, and you kind of figure out like how much can you put into one manuscript. It may be that you can only put the qualitative work into the first manuscript because to do that part justice is a full manuscript, or vice versa for the quantitative piece. I guess that's, I think it's very study specific how you present those findings, and you just don't want to give short shrift to any part of it.